Hello students, welcome to my channel. We are discussing the important questions of chapter number 11 and this is video 3 of the series. If you haven't watched the initial two videos, please do have a look. PCR was discovered by Carey Mullis. So if a question on PCR comes, what is PCR? Definitely your first line should be that it was discovered by Carey Mullis. Second question is DNA fragments move at different distances in gel electrophoresis. So they move because their molecular weights are different. You have seen that the larger ones, they are always near the negative terminal and the smaller ones, they are always towards the positive terminal. DNA can be isolated from fungi using chitinase because chitinase enzyme breaks the chitin and chitin is a component of its cell wall. Ethidium bromide, which was used in the process of gel electrophoresis, the next question is based upon that. So ETBR means they are asking about ethidium bromide fluorescences at the wavelength. So this is going to be the wavelength 254 because this is associated with UV light. Okay. So while a question on gel electrophoresis come, you can also use the same wavelength in one of the sentences of your answer. The ability to form multiple copies of antibiotic resistant gene, of course it is known as transformation because E. coli was otherwise not carrying any antibiotic resistant gene. So by the process of biotechnology it was. So this actually process, the last process where we introduce and it expresses, this is known as transformation. DNA is amplified by PCR and it can be used to produce multiple copies of DNA. Next question is, name the polymerase that is generally used in PCR. So it is Stack polymerase. And what is the source of this enzyme? So it is being taken from the thermophilic bacteria. Why are we saying thermophilic bacteria? Because this is one bacteria which can bear high temperature, because denaturation, say it occurs at 92 degrees Celsius and then also no denaturation occurs. So what is the source? Thermus aquaticus is the source of this bacteria. You can also write in line it is thermophilic because it can grow at high temperature, which is being favored by the denaturation step of the PCR. Next question, why is the enzyme cellulase is used for isolating genetic material from the plant cells but not from the animal cells? Very easy question. Because only plant cell contains the cell wall that is made up of cellulose, but animal cells do not contain any cell wall. And cellulase enzyme basically acts upon the cellulose which is missing in the animal cell. So that's why whenever we want to extract DNA from the plant cell, cellulase has to be used which will break the cell wall. Next question, starting from double-stranded DNA suggests a strategy for obtaining large amount of pure single-stranded DNA for the sequencing process. Here in this question, this is the key term, obtaining large amount of. So whenever you see it, please do quickly correlate that they are asking about PCR. The link to PCR will be given in the I button. If it is not clear to you, you can click on that video and get your concepts cleared. How can the following be possible for biotechnology experiments? Again, for this question, isolation of DNA of bacterial cell, I'm not going to describe here, because there is already a recorded video on it. So for this also I'll be sharing the link in the I button. And reintroduction of recombinant DNA into a bacterial cell, means they are asking about transformation process here. So for this also I'll be sharing the link in the I button. Give the characteristic feature and source organism of the DNA polymerase used in PCR. Characteristic feature we have just discussed in question number 7. That this DNA polymerase from Thermus aquaticus has the capacity to bear high temperature even of 92 degrees Celsius, which is being taken in the denaturation step of PCR. So source organism is Thermus aquaticus only. Question number 12 is, what is the principle of gel electrophoresis? Name the compound which is used for staining and what is the color of such stained DNA. Principle of gel electrophoresis is that the DNA fragments, they get separated based upon the molecular weight. The smaller ones will be moving faster and they would be found at the positive terminal, and the negative ones would be the DNA fragments that would have more molecular weight. They will be found near the negative terminal. So principle is that the fragments of DNA will be isolated based upon their molecular weight. Name the compound which is being used for this staining. So it is ethidium bromide. There was just a question on ethidium bromide in MCQ that we discussed. What is the color? It is orange. Okay. Describe briefly the following, which is bioreactor. So for this also I'll be sharing a link in the I button. You can go through it. Question number 14 is, what was the experiment of Cohen and Bohr? You remember it is being given in the second page of chapter number 11. So for this also I'll be sharing a link in the I button. You should know how they progressed with their experiments. This is one of the NCERT questions which is being overlooked by many of the students. So I will definitely suggest that please do learn the names of few recombinant proteins because it will be helpful to you, right? So I am just showing you few. You can pick up any one of your choice. You have already studied about humulin, so you can pick up this one also. Blood clotting factors 8 and 9. This will be helpful to learn because you have to appear for NEET shortly. So this will definitely help you out. Tissue plasminogen activator. It is used for myocardial infarction. Interferon alpha is used for hepatitis C. Interferon beta is used for multiple sclerosis. And interferon gamma is being used for granulomatous diseases. Okay. Here there is a spelling mistake, so you can make it correct. Learn a few recombinant proteins. It will definitely help you out. At least 5 should definitely be learned. Stay connected for more videos. Thank you.